Hello, I'm Matthew Cooper, Director of Kidney and Pancreas Transplantation at MedStar Georgetown Transplant Institute and Professor of Surgery at Georgetown University School of Medicine in Washington, D.C. Thank you for your interest in learning more about FCR001, an investigational cell therapy that is being developed by Talaris Therapeutics. FCR001 is designed to induce immune tolerance, and it is being investigated for use in conjunction with living donor kidney transplant. Today, I would like to introduce you to FREEDOM-1, the Phase 3 clinical trial of FCR001 in living donor kidney transplant recipients. FREEDOM-1 is a randomized, controlled, multicenter Phase 3 study of safety and efficacy of FCR001 versus standard of care immunosuppression in de novo living donor kidney transplant recipients and safety in FCR001 donors. The primary endpoint is proportion of FCR001 recipients who are free from immunosuppression and without biopsy-proven acute rejection at 24 months post-transplant. A key secondary endpoint is change in renal function in FCR001 recipients as measured by eGFR from post-transplant baseline at month 1 to month 24. In FREEDOM-1, donors will undergo stem cell mobilization and apheresis for FCR001 processing. Prior to transplant, FCR001 recipients will undergo mobilization, collection, and stem cell cryopreservation. FCR001 recipients will undergo conditioning with fludarabine on days -4 to -2 and will receive cyclophosphamide with Mesna on day -3. Dialysis will be performed after each conditioning treatment. On day -1, recipients will receive low-dose total body irradiation. FCR001 recipients will start immunosuppression with tacrolimus and mycophenolate mofetil, or MMF, 3 days prior to kidney transplant. Stem cell transplant via FCR001 infusion occurs 1 day after kidney transplant. Recipients will receive an additional dose of cyclophosphamide plus Mesna on each day on post-transplant days 3 and 4. These doses of cyclophosphamide are intended to further mitigate the risk of GVHD. And to support the expected hematologic nadir, recipients also begin daily doses of G-CSF on day 5, which continue until absolute neutrophil count is greater than 1,000 cells per microliter 3 days in a row. Patients who have stable donor chimerism, meaning at least 50% donor T cells, and stable graft function with no biopsy-proven acute rejection or GVHD, may discontinue MMF at the end of 6 months post-transplant, and may be able to wean tacrolimus from month 9 to month 12. In the standard of care arm, donors undergo nephrectomy on the day of transplant. Recipients will be placed on immunosuppression after transplant according to a standard of care protocol. Recipients in both study arms will be followed for 5 years post-transplant. Recipients in FREEDOM-1 must be at least 18 years of age and undergoing a first or second kidney transplant from a living unrelated or related donor who is not an identical twin. Donors must be 18 to 60 years of age and willing to undergo mobilization, apheresis, and 12-month safety follow-up. Exclusion criteria for recipients include panel reactive antibody greater than 80%, ABO incompatibility, and presence of donor-specific antibody. Male recipients of a biologically unrelated female donor kidney are also excluded. Exclusion criteria for donors and recipients include history of malignancy within 5 years of screening and known bone marrow aplasia. Clinical updates for FREEDOM-1 may be provided periodically on the Talaris Therapeutics website. And in the meantime, if you would like to learn more about FREEDOM-1, please visit the study site at freedom1study.com or the NIH clinical trials site. Thank you for your time and your commitment to kidney transplant.